Hello. I'm Marie Loescher. I'm a PhD student at Catherine Talon-Baudry's lab and co-supervised by Patrick Haggard. And I'm going to talk to you about how signals from the body interact with perception. And I'm going to actually talk about the heart, so sorry about that. So all of us know by now just how much information our brain constantly receives from our body. And knowing that, we might ask the question of how these so-called interoceptive signals interact with the information that comes from the outside world through our senses, such as audition, what we call exteroception. In other words, how does the body influence perception? And interestingly, there seems to be a fundamental but implicit split in the literature so far, where on the one hand, we have the competition team, or the proponents of a general principle of competition between signals from the inside and signals from the outside. So in this view, bodily signals, or interoceptive signals, inform the brain about the body in about the same way as signals from our senses inform the brain about the world. And depending on context, task, and maybe some intrinsic alternation between the two, the brain would allocate more neural resources to either the one or the other. So a sort of trade-off idea. On the other hand, we could also assume that interoceptive signals could facilitate at least some aspects of exteroception, and in particular, maybe those linked to the subjective aspects of perception. So this comes from a bit of a philosophical idea that every subjective experience presupposes a subject, so some sort of very basic minimal self. Philosophers have called this minimal self. And one hypothesis under this facilitation framework in particular is that maybe some bodily signals, such as the continuous beatings of the heart, actually signal or underlie this minimal self, and that maybe it is the integration of these bodily signals with the exteroceptive signals from outside that then yields subjective conscious perception. So we will actually only look at the heart for this now. And the heart is interesting because together with the stomach, it's one of the only organs that actually produce their own intrinsic electrical activity. And this electrical activity is monitored by the brain at all times, unconsciously, and generates what is known and what we by now all know as the heartbeat-evoked response. And this heartbeat-evoked response has actually been measured in quite a wide array of regions in the human brain, including some primary sensory cortices. And the amplitude of this heartbeat-evoked response has been classically taken as a marker of a neural processing allocated to the heartbeat. So how much does the brain actually care about the heart in this moment? And quite a lot of research exists on the heart already. For instance, we have two studies by one of our tutors at the summer school that just went by, Ezra Al, who showed that the amplitude of heartbeat responses would negatively predict whether a tactile stimulus was going to be perceived or not. And this was, of course, interpreted as evidence for the competition account. Under the facilitation framework, we have a study by Park and colleagues from my lab, which actually showed that the amplitude of heartbeat responses positively predicted whether a faint visual stimulus was going to be perceived or not. And then we also have a whole host of studies that linked the heartbeat responses to self-related cognitive processes in a variety of cognitive domains, including many studies from my own lab, but also from others. So we see that the evidence so far is a bit dispersed, a bit contradictory, and the frameworks are kind of implicitly assumed, but never really actually tested, both in the same paradigm. And so that is what we actually wanted to do with this project, was to, for the first time, really directly test both accounts and their predictions. So to test the competition account, we just need some sensory stimulus that they were going to perceive or not, so that's rather easy to test. But to properly test the facilitation account, we would actually need some sort of manipulation linked to the self in this perception. And the self is a bit tricky of a concept to target. So what we came up with was to manipulate the relevance of a stimulus for the subject. So to do that, we made use of a well-known space around the body, which is called the peripersonal space. It's kind of like really just the space close to your body. And in this space, stimuli seem much more relevant to you than when it's further away. If you think, for instance, of someone standing way too close to you, it's really hard to ignore. And we know behaviorally there's many effects of this peripersonal space. And one of the effects is that participants are faster responding to stimuli when they're presented close to the body than when presented further away. And this effect is also modulated by many other factors, such as the stimulus valence, whether it's reachable or not, the motion trajectory, whether it's likely to hit the subject or not. So all of this suggests and has been suggested so far in the peripersonal space literature that it kind of reflects a space of self-relevance, like a bubble of heightened self-relevance around you. So that is what the manipulation that we then used for our task. And the way that we did that was to use a well-validated paradigm by Ronga and colleagues, where subjects respond to a faint tactile stimulus on their hand as fast as possible. And while they receive the faint electrical shocks, there is a short beep that's being concurrently played from one of two speakers, either a speaker that is placed very close to the stimulated hand or one that is about 1.5 meters away from it. And the subjects are always instructed to just respond to the touches and to the tactile stimuli and to just ignore the sounds. And so these audio-tactile trials are randomly intermingled with trials in which only a sound is presented, the catch trials, or trials in which only the tactile stimulus is presented, which kind of serves as baseline. And so what we see behaviorally is what was already found before, and we replicate this paradigm where we see that the subjects are faster responding to the tactile stimulus when the concurrent sound is presented close to the hand compared to when it's presented far from the hand in the audio-tactile far condition. And we can also see that this effect, so we model this with a linear mixed effect model, and we can see that this effect is small but pretty consistent across all our 37 participants. So yeah, that's what we wanted. And so with this task, we now have a task where on the one hand, we can just probe simple tactile processing by measuring tactile reaction times. But we also have this interesting distance effect, which is then our self-relevance effect. And with that, we can manipulate self-relevance. So how does all of this now interact with the heart? So to test that, we measure EEG and also the electrocardiogram, and then we will look at the last heartbeat evoked response before stimulation onset. And then we can model trial by trial heartbeat evoked responses, reaction times, and this self-relevance effect, which is this interaction between reaction time and sound distance, in a common GLM. So if cardiac and tactile stimuli compete for neural resources, then we should see that a large pre-stimulus heartbeat evoked response predicts slower tactile reaction time. So that corresponds to a main effect of reaction times in our GLM. And in fact, that is what we find. So in this cluster over here, between 450 and approximately 500 milliseconds after the cardiac hour peak, we can see that actually the amplitude of heartbeat evoked responses will predict whether reaction times are slow or fast. And if we plot the heartbeat evoked response across these electrodes separately for slow and fast trials, we can see that actually in this significant time period here, higher heartbeat evoked response amplitudes will correlate with slower reaction times. So this is really in line with what the competition account would predict. So Ezra will be very happy about that. On the other hand, if the neural processing of cardiac signals is one of the mechanisms underlying the subjective aspects, self-related aspects of perception, then we should see that a large pre-stimulus heartbeat evoked response also predicts a strong self-relevance effect. And in fact, also for the facilitation account, the predictions are confirmed. We first of all find that in this frontal cluster, the heartbeat evoked response amplitude will predict the strength of the self-relevance effect. And again, if we plot the heartbeat evoked response across these electrodes, we can see that well, this time it's a negative amplitude, but the higher the amplitude, the absolute amplitude, the stronger the self-relevance effect was for this given trial. Interestingly, these two effects seem to be fairly independent from one another. So first of all, there is a time overlap, but also a little bit of a difference, so it's a different timing. Second, it seems to be driven by different brain regions. So the competition effect, so the main effect of reaction time, is mostly driven by an area that's basically in the posterior somatosensory cortex. Whereas the facilitation effect was driven by three different sources, one here in the frontal, one is inferior temporal, and this is in the anterior cingulate cortex. And lastly, there doesn't seem to be any trade-off between these two. So you could think that, okay, well, maybe it's just a trade-off, either there's competition or there's facilitation. But no, we can see that if we quantify the effect, so basically the average effect size per subject and plot one against the other, there is really no anti-correlation whatsoever and no correlation for that matter either. So that is pretty cool. And then, of course, we ran all of the controls that we have to run to make sure that actually there is no difference in the cardiac parameters between the different conditions, and that the effect is truly time-locked to the neural response to heartbeats. And also there was no difference in arousal parameters such as pupil diameter between audiotactile near and far. And lastly, we also investigated whether this effect was also reflected not only in reaction times, but also in the stimulus evoked potential. And again, we find the same pattern. So heartbeat evoked response amplitudes predicted simple somatosensory evoked potential amplitude, but also the effect of sound distance on somatosensory evoked amplitude. Voilà, that's it, more or less. So conclusions are that for the first time, we could simultaneously test competition and facilitation accounts prediction in the same paradigm. And we find a confirmation of both. So on the one hand, we saw that the neuroprocessing of heartbeats impedes tactile perception. And on the other hand, we can also see that neuroprocessing of heartbeats in other regions facilitates some other aspects, and we think it's related to subjective aspects of perception. And also we find that these two effects are probably reflecting different underlying mechanisms. And with that, I thank my two supervisors, my team, and you for your attention.
We have time for one question.
Thanks, that was a super cool talk and analysis and task. I really like how you presented it with the competition and facilitation hypothesis. I was just wondering, was there any effects of how close the previous heart evoked potential was to the next part of the task, essentially?
No, there wasn't.
Oh.
And also we repeated the analysis with taking both the second to last and the last heartbeat evoked response before stimulus onset, and it doesn't seem to make that big of a difference. Same effects reproduce, yeah.
Thank you.
Sorry, we are running out of time. So
Oh,
thank
sorry.
you. Thank you, Marie.
Thanks.